Hey Beaklings, it's Mr. Boylan, and today I'd like to talk to you about mitosis, which is M phase of the cell cycle. We're going to talk about how the nucleus divides and how the rest of the cytoplasm divides via cytokinesis at the same time. Okay, so here we go. We've already learned about the cell cycle, and as you can see, most of these pictures here are in some sort or some phase of cell division. This guy right here is probably a late G2 interphase cell. And then we go into Prophase, Prometaphase, Metaphase, Anaphase, and Telophase. You should really feel good enough about what we do today that you could just look at these cells and immediately know what phase that you're in. Okay, so here we go. Mitosis. So we're going to start in interphase. So we're just probably at G2. We're just getting ready to divide, but we're not quite in M phase just yet. All right, let's take a look.
The cytoplasm of a cell in late interphase contains two centrosomes, each of which may contain a pair of centrioles. In the nucleus, the chromosomes have been replicated during S phase, but are still dispersed in the form of chromatin. One or more nucleoli are present.
Okay, so interphase, pretty simple. The cell is still a factory. But now we reach that point where we're going to go into cell division. So we're crossing the barrier from interphase, we hit that threshold of proteins, and we're heading into M phase. Okay, so what the first part of M phase is going to be called prophase. Okay, now I'm going to have a separate video where we draw all of these phases together, but eventually you'll see that the hallmark of prophase is definitely the coiling up of chromosomes. So let's watch the video first.
During prophase, the nucleoli disappear and chromatin fibers coil up to become discrete chromosomes. Each chromosome consists of two identical sister chromatids joined at the centromere. Microtubules grow out from the centrosomes, initiating formation of the mitotic spindle.
Okay, so as you can see, there is an early spindle forming here. Those are those microtubules that we talked about. Only in this case, they're not part of cilia or flagella. They're not tracks for organelle movement. These are literally going to help us rip our chromosomes in half in our cell. And they come out of the centrosomes, which have these centriole pairs inside of them. So if we go back here, we can take a look. Okay. The chromosomes are starting to coil up. I'll teach you how that happens. And you can see that the spindle's starting to form. The nucleoli have disappeared. Okay. Now, most textbooks would call this early prophase and this late prophase. Your textbook calls this prometaphase. Okay, so it's just a little later, and as you can see, the nucleus is pretty much gone. The spindle's formed and grabbing the chromosome. So let's take a look at the next phase, prometaphase. Okay, and like I said, you can just consider this late prophase.
During prometaphase, the nuclear envelope breaks into fragments. Some of the spindle fibers reach the chromosomes and attach to kinetochores, structures made of proteins and specific sections of DNA at the centromeres. Non-kinetochore microtubules overlap with those coming from the opposite pole.
Okay, so like we said, in prometaphase, the nuclear envelope is pretty much gone. You have these little kinetochore proteins on the outside of the centromere, okay, which is where your chromosome connects, don't forget. So the spindle fibers will grab that kinetochore, okay, and they're going to start moving the chromosomes toward the center of the cell. Okay, so that was prometaphase. So we're two phases in the M phase at this point. We went through prophase and prometaphase, and now it's time for the next phase. We're going into metaphase. And as you can see here, we're really just going to line the chromosomes up down the center of the cell. This is a pretty easy and short phase. Let's have a look.
At metaphase, the mitotic spindle is fully formed, and the microtubules attached to kinetochores move the chromosomes to the metaphase plate, an imaginary plane equidistant from the poles.
Okay, so you might know why we did that already, right? The whole point of doing that is so we can now, we went through S phase, and S phase was when we had a chromosome, we doubled it, so that they can become sister chromatids. And now that they're lined up down the center of the cell, we can pull them apart. Okay, so metaphase is very important that we line up these chromosomes right down the middle. And that'll bring us to our next phase, which is anaphase. And something hugely important happens in anaphase. Okay, so let's take a look. We're actually going to double the number of chromosomes in the cell here. Okay, and that's something that's really difficult to understand at first because we have 46 chromosomes right now in your cell, but when you split them, technically, one chromosome with two sister chromatids separates to become two full-fledged daughter chromosomes. And I know that's just semantics, but that's super important for you to understand that this chromosome right here one chromosome becomes two chromosomes. These are no longer called sister chromatids once you separate at the centromere. All right, so let's give a look to anaphase and see what's going on here.
Anaphase begins when the two centromeres of each chromosome come apart, separating the sister chromatids. Once separate, each sister chromatid is considered a full-fledged daughter chromosome. Motor proteins of the kinetochores walk the daughter chromosomes along the spindle microtubules toward opposite poles, and the microtubules shorten. At the same time, the spindle microtubules not attached to chromosomes lengthen, pushing the two poles farther apart and elongating the cell.
Okay, so a lot happening right here. Did you notice the name change? You could rewind the video if you have to. This was a chromosome, but the second these two sister chromatids came apart, which is actually, this is the beginning of anaphase. We're in metaphase right now, lined up down the center, metaphase, metaphase, metaphase. The second they go like this and separate, okay, that's anaphase. And if you notice in the video, they changed the name from sister chromatid to daughter chromosome. The other thing that's important, if you look at the video here, is the non-kinetochore microtubules, the ones that aren't connected to the chromosomes, they're still lengthening. And when they hit into each other, that actually elongates the cell. Technically, we kind of have started a process called cytokinesis here. Okay, so let's go ahead and finish up cell division, or M phase, okay, by looking at telophase here. And telophase is kind of the opposite of prophase, right? In prophase, don't forget what happened. The chromosomes coiled up. You went from chromatin, okay, to chromosomes, and the nuclear envelope disappeared, right? The nucleolus disappeared. Well, now we're going to have to remake all of that stuff and uncoil back into chromatin fibers. So let's take a look.
During telophase, nuclear envelopes form around the identical sets of chromosomes at the two poles of the cell. The chromosomes uncoil, and nucleoli appear in the two new nuclei. Meanwhile, cytokinesis begins, splitting the cytoplasm and separating the two daughter cells.
Okay, so in telophase, you finished up mitosis. Mitosis is the division of the nucleus. So at this point, the DNA is already separated. You're starting to form new nuclei around it. That's mitosis. But also, if you noticed, you have a cleavage furrow, and cytokinesis happened, right? You pinch these actin filaments and pop your cell in half. Okay, so technically, both of these little cells would now be G1 daughter cells, right? And they're going to go through the whole cell cycle all over again. Okay, so that's pretty much mitosis. It takes a long time. You have to go through it again and again and again, but eventually you'll understand it and be able to recognize those phases. Let's practice here. This is a new lung cell, and hopefully we'll be able to see the phases of mitosis as it divides here. Okay, so I think I can see chromosomes in there, right? So I believe that I can see, yeah, this is probably prophase, okay? And actually, it looks like I don't really see a distinct nucleus. It looks like prometaphase, right? Because I don't see the spindle, but those are moving around. And the fact that those chromosomes are moving around means, okay, yeah, they're fully coiled now. The spindle's probably pushing them around. And as you know, they're probably heading towards the center of the cell, right? And what's going to happen once they all get in the center, I'm just going to fast forward this a bit. Look at this. Now they're all going to the center. I would say that's probably metaphase right there, right? Looks like they're down the center of the cell. And you know what comes next is we're going to double the number of chromosomes because now the sister chromatids are called daughter chromosomes. Let's go ahead and split them in anaphase. Here we go. You see that? Right there's the beginning of anaphase, right? Metaphase, anaphase. And in anaphase, they're going to move towards the poles of the cell. And look what happens in the middle. We're starting to form a cleavage furrow. We're probably in the telophase right now, right? Because look at the chromosome strands. Look at how they disappear. We're going from chromosomes back into chromatin, right? Like those thick spaghetti noodles of DNA and protein. So let's go through it one more time. So prophase, prometaphase, right, for sure. Metaphase is pretty quick. Once they're all in the middle, that's just called metaphase. And then anaphase, telophase, and cytokinesis. Right? Pretty cool stuff. Okay. Now, how do you fit all of that DNA, okay, which is like gigantic amounts of spaghetti, if you want to think of it that way. So DNA is a double helix. It doesn't exist in your cell as a double helix. A double helix, let's say, is about the size of a fishing string, but chromatin fibers are big, right? That's DNA wrapped around these things called histones, which form nucleosomes, and it gets to be this thick fiber. So point is, you have chromatin, but you have 46, or after S phase, 92 pieces of chromatin. Imagine like a plate full of spaghetti with gigantic pieces of spaghetti and 92 of them. And I said, hey, do me a favor, quick separate those evenly and perfectly. Quickly, please. Wow, that's hard to do, right? So we have to have a really complex way to organize all of these chromosomes, okay? And this has taken billions of years of evolution to get just right. But let's see how our chromosomes coil up.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing.
Pretty amazing
At other
stuff there,
times,
right?
as we can see here at the end of cell division, our DNA becomes less highly organized.
Okay, so that's how we can do this efficiently, right? We coil it up so specifically like that so you can, and then that's when you'll see it, that's prophase, so that you can attach it to microtubule fibers at the kinetochores, move it to the center of the cell, and split it apart, and then have it go back into each side of the new daughter cells, uncoil back into chromatin, and there you go. You got a G1 cell, and you made a perfect genetically identical copy. Okay, so that cytokinesis is a little interesting that happened there at the end. In an animal cell, it's pretty easy to do because we just form a cleavage furrow. Animal cells can literally just pinch in half thanks to those actin fibers or microfilaments. Plant cells have it a little bit trickier though because they have a cell wall, so they have to form something called a cell plate. Okay, so let's see, because remember the definition for cytokinesis is really this division of the cytoplasm.
In animal cells, cytokinesis begins with the formation of a cleavage furrow. At the site of the furrow, a ring of microfilaments contracts, much like the pulling of drawstrings. The cell is pinched in two, creating 2 identical daughter cells. In plant cells, cytokinesis begins when vesicles containing cell wall material collect in the middle of the cell. The vesicles fuse, forming a large sac called the cell plate. The cell plate grows outward until its membrane fuses with the plasma membrane, separating the 2 daughter cells. The cell plate's contents join the parental cell wall. The result is 2 daughter cells, each bounded by its own continuous plasma membrane and cell wall.
Okay, so there you have it. That's cytokinesis, and that is a background on mitosis. I hope that was helpful. My advice to you, though, is that's not deep enough. So what I think you should do now is I think you should go check out my drawing or someone's drawing video and double check that you can actually draw the phases of mitosis. And that way you can really think about it while you're drawing them and maybe make some little notes or annotations to yourself. But it's a really cool process, and it's a very simple process once you study it. Now, one last piece of advice, I would definitely get this down as best you can, because eventually we're going to talk about making sex cells. We're not talking about a cell cycle now, we're actually talking about making gametes, which are sperm and eggs for a human. If you understand mitosis really well, meiosis, which is quite a bit more complex, meiosis will be a breeze for you. But you really have to go in there with a firm footing of mitosis first, okay? So I hope that was helpful, and I will see you guys later. Take care.